Enzymes are proteins that act as biologicals and catalysts which accelerating chemical reaction by lowering down the activation energy to speed up chemical reaction. So now we are going to show some example of method that enzyme and substrate used.
Hi guys, what are you doing?
We are studying biochemistry for finite examination. Let's join us.
Actually, I don't understand about this topic. This topic, I know about enzyme. Let's study it together.
Hey, I have a 3D model for the enzyme. Oh really? That's nice. So we can properly explain about enzyme.
Let me introduce about this model. This is a substrate and this is enzyme. When the substrate bind the active site of the enzyme will form enzyme substrate complex.
That's right. So when the substrate bind to the active site, there is a stress applied, and it will break down the substrate and produce a product. The enzyme is free to bind with other substrate.
Let me show. This is a substrate bind at the site of the enzyme with the form enzyme substrate complex and produce product.
I think I know the second one. E2. The inhibitors is a substance that will slow down the rate of reaction. There are two types of inhibitors. The first one is competitive inhibitors. Competitive inhibitors will block the substrate from binding to the active site at the same space. So, due to the competitive inhibitor have the similar shape like the substrate, so it will bind to the same space, the active site and the enzyme.
There is another type of enzyme inhibitors, which is non-competitive inhibitors that exhibit allosteric regulation. It can be distinguished by the binding of inhibitors to an allosteric site, which is here. The allosteric site is different from the active site. When the inhibitors binding to allosteric site, this will decrease the efficiency of the enzyme.
How about the last one, hand in glove model?
Actually, we don't really know about that part.
How about others?
Can you guys keep it down, please? I can concentrate.
Well, we study enzyme, but we don't know the last part.
That's easy. Let me explain.
In this free model is the changing shape of the active site of the enzyme when binding with the substrate to achieve optimal speed. The initial shape of the active site of the enzyme are not exactly complementary with the substrate. However, once the substrate binds to the active site of the enzyme, the active site of enzyme will change and adjust its shape to complementary with the substrate. As you can see the hand in the model, where the shape of the glove is made to perfectly fit the hand. It is molded around the hand, even though the glove and the hand is broadly comparable in shapes.
Wow! Get it!
After performing role play, we can conclude that enzymes are specialized proteins that act as catalysts, accelerating biochemical reaction. So that's all. Thank you.